Good Tuesday evening, everyone, and welcome to Shoals Marine Laboratory's weekly marine seminar series. I'm Dr. Jennifer Seavey, the Executive Director of Shoals. For those of you who are not familiar with us, we are the largest and the oldest undergraduate-focused marine lab in the country. We are an institution of both UNH, University of New Hampshire, and Cornell, and are located on Appledore Island, which is in the Isles of Shoals, about 10 miles offshore of Portsmouth, New Hampshire. This series, which will run through the end of 2020, is designed to bring the Shoals community together to learn and discuss and develop new ideas for marine science and conservation. So tonight, our format, as usual, is a 45-minute talk, followed by 15 minutes Q&A. And if you want to ask a question, which we highly encourage, please use the Q&A box down there at the bottom of your screen. Please type in your question and we'll get to them at the end. And if you have any technical needs, also use that box and we will help you out. So tonight, I'm really excited that we have Dr. Liz Harvey with us. She is a biological oceanographer interested in marine microbial dynamics. Her work involves an integrated approach across multiple spatial scales from molecules to ecosystems to recognize the mechanics underlying shifts in phytoplankton population dynamics and primary productivity. And ultimately, she's interested in trying to understand biogeochemical cycles and food web structure in the ocean. She has an undergraduate degree from just up north of us, University of Maine, a master's degree from Western Washington University, so she travels around, and a PhD from the University of Rhode Island, where we have lots of Shoals alumni there. And before joining UNH, which she did just last year, she was faculty at University of Georgia for 4 years. So tonight, she definitely wins the prize for the greatest title, and she's going to be talking to us about microbial mortality and the ocean. So welcome, Liz. We're happy to have you here. I'm going to hand you the virtual floor, if you will.
Thank you very much for inviting me. I'm really excited to chat tonight. Let me share my screen. Hopefully everyone can see that. Thanks all for joining in this evening. I hope to provide you with an uplifting talk. No one warns you when you go into choosing what you study for your science that if you study microbial mortality, that might not be like a super awesome thing to talk about in the middle of a pandemic, but I think we can make it interesting and exciting. I just want to give a shout out to the artist of this drawing right here, or painting, Christiane Baker. Apparently she is also a Shoals alum that I just learned, but she does beautiful, beautiful marine art. She has a glass installation at Bigelow Lab in Maine, and I really appreciate all of her phytoplankton work. So today we are going to talk about microbial mortality in the ocean. And to start, I want to sort of lay the landscape a little bit of what exactly we're talking about. So if you went out and you went to your internet browser, whatever you choose, we'll say Google, and you typed in ocean, and then you hit the little image button, you get all of these beautiful pictures of the ocean. They're amazing, they're wonderful, probably lots of tropical images all around the world. But one thing that search would have in common is this sort of view of the ocean, right? The perspective of the ocean from the view of a human, right? We look out and gaze over the ocean and it's beautiful, and we recreate in the ocean, and we just sort of all have a affinity for it. But I think a lot of times what's lost is what happens actually underneath the water. And from my perspective, that's where all the super interesting stuff happens. So this is a image of the ocean that I like to think about. This is just another painting that sort of depicts the diversity of organisms that live in the planktonic or oceanic environment. Now, to be really clear, these are not all to scale, right? Like it would be super awesome, but we don't necessarily see giant copepods that are the same size as larval fish that swim around in the ocean. But the point of this image is to give you an idea of the vast diversity of microbes that live in the water column in the ocean. And so I like to spend my days thinking about what are they all doing there together? How do they interact with one another? And then what are the consequences of those interactions? And I would be very remiss if I didn't add to this painting both bacteria and viruses, because they are very important players, very important microbes in the marine environment. And they actually play very important roles in interacting with other phytoplankton and larger organisms. So we need to make sure we include those in our sort of view of underwater diversity here. So how do we calculate how phytoplankton abundance or any microbial abundance changes in the ocean? And we're going to use, I promise this is the only equation in the talk. It's a very simple equation, and we'll sort of walk through it. So phytoplankton or microbial biomass at any given time is really based on how much you have there to start, so the sort of initial biomass that you have, multiplied by a net growth rate, how fast are things growing, and then by some unit of time. And you can multiply those together, and because microbes grow exponentially, you have that little e in here for the exponent, and you can project or calculate how phytoplankton or microbial biomass will change over time. When you look at the equation even sort of further, you can delve down into this net growth rate. And the net growth rate is really the sum of things or processes that stimulate growth. So how do we get microbes to grow faster? And then also processes that contribute to their loss or their death. And you really need to balance both of those, or understand and quantify both of those, in order to get a really accurate net growth rate. So if we're thinking about phytoplankton, which I'm going to focus most of this talk on phytoplankton, when we think about growth rate anyway, light and nutrients are really essential for phytoplankton growth. So just like, you know, you can spray Miracle-Gro over your garden, phytoplankton really want all the nitrogen and phosphorus you can give them. They also really want light to be able to grow. And if you change the formulation of those different things, then you'll change the rate at which they grow. So less nutrients and light, slower growth, more faster growth. And that's pretty well established. We know a lot now about the different nutrients that phytoplankton need, their different requirements, their different light regime requirements, but a lot less is known about how phytoplankton die or how they're lost from the system. So the two main ways that phytoplankton can be lost from the system is due to physical processes. So they get, you know, maybe a storm comes through and really mixes up the phytoplankton and moves them physically to another location. But they can also die, and that's what we're going to focus on today. So my research really looks at how interactions between microbes impact these loss rates and these loss processes. Okay, so how do things die in the ocean? There's actually quite a few ways that microbes can die. And I apologize in advance for anyone who has not caught up on Game of Thrones. There will be maybe like a 30-second Game of Thrones spoiler that's coming right up. If you haven't watched it, mute me for a second and come back in a second. But I like to sort of think about, maybe I anthropomorphize my organisms a bit too much, but I really enjoy thinking about how they interact with one another. And in terms of death, there's lots of ways to die, very similar to how were many ways to die in the show Game of Thrones. So if you're familiar with that show, these are some of the notable deaths that occurred throughout the many seasons of the show. And not one death was really the same. And really the interesting thing is that each time a major character died in the show, there were significant ripple effects throughout the show that changed the plot line or changed directions of characters. And very similar things happen in the ocean. So we can sort of think about all the different ways that something can die in the ocean, and then how it can have a ripple effect on how nutrients and carbon and other things that we're interested in are cycled throughout the ocean. So I really like this image. This is an image from a review paper by Alexandra Warden, and it sort of details these different ways that phytoplankton especially can die. So they can be grazed. So predation is probably the largest sort of avenue of loss for phytoplankton in the ocean, and we'll talk a little bit about that today. Parasitism is also an important way. Sapotrophy, mixotrophy, meaning organisms can both conduct photosynthesis and consume carbon. You have bacteria phytoplankton interactions. That's what is meant here by the phycosphere. And then you can have symbiotic interactions. Sometimes those symbiotic interactions turn bad. So sort of like a relationship gone bad. You can have these very nice symbiotic relationships in the beginning, and then something happens and it can cause the phytoplankton to die. And then this very tangled food web down here is trying to show where the carbon in these interactions, the resultant carbon is moved, right? So if you are a phytoplankton and you get sort of infected with a parasite or a virus, there's no real way for you to recover from that. So you're going to die, and all of your delicious nutrients are going to sort of burst out and become dissolved organic material that bacteria will use, remineralize into nutrients that are usable by the new phytoplankton that are growing. So you sort of stay in this, what's called the microbial loop. And that's a different pathway than if you were a phytoplankton that was eaten by a heterotrophic protist or by a grazer. So if you're eaten by something, your carbon now moves up the food web, not down into the microbial web. You're going up now into higher trophic levels. So those protists will get eaten by zooplankton, which will eventually then get eaten by fish and get eaten by, other large, more macrofauna or macro vertebrates that we all sort of enjoy in the ocean. And so there's these two very distinct pathways, and we don't quite understand how often phytoplankton go through virolysis versus grazing or any of these other different ways of dying. And so that's sort of the root of my research. So today we're going to talk about, we have two stories we're going to talk about, both, different pathways sort of on that, that food web model. First, we're going to talk about microzooplankton grazing, and then we're going to talk about parasites, which is something that I'm newly interested in, but really excited about. And I think it sort of freaks my mom out when I tell her that I am really excited about parasites, but they are very exciting in the ocean. Okay, so the first story is about microzooplankton grazing. For those that don't know, microzooplankton are single-cell grazers of phytoplankton. So in this image here, this really large cell here is a microzooplankton called a ciliate. They have cilia here at the end of their body, and it is going to eat, hopefully, this delicious small phytoplankton cell here. Microzooplankton grazing accounts for about 60 to 70% of daily primary production loss. So they consume about 60 to 70% of all the primary production that is produced on a daily basis. So they're very important in phytoplankton loss processes. And if this isn't quite intuitive on how this relationship works, this is a very analogous relationship. So here the poor leopard seal is just about to eat this penguin. This is exactly what's happening over here on the left. It's just that it's on the microbial level. And so just as this interaction is cool and fascinating, I think this interaction is really cool and fascinating as well. So today we're gonna think about a story with a beautiful phytoplankton species called Emiliania huxleyi. Amelia, or ehux, as you'll probably hear me call it, is what we call a coccolithophore. And a coccolithophore is a type of phytoplankton that is basically surrounded by calcium carbonate liths. So they essentially have all these little plates of armor around their shell. And these calcium carbonate liths are essentially chalk, right? And so they're white. We can really easily see them by satellite. So here's a picture of a giant E. huxleyi bloom in the Barents Sea in Norway. And you can see that this bloom is huge. It really reaches quite a large geographic extent. And this is the result of a pretty dense bloom. All the liths are falling off and being very reflective from satellite. We just had blooms of E. hux this summer, all around Norway, also around in the UK. If anyone has been to the White Cliffs of Dover, the White Cliffs of Dover in the UK is really made up of ancient Coccolithophore skeletons, essentially. And they're very, very important in the ocean in terms of carbon cycling and also in terms of the cycling of sulfur. So they're an important organism, and we're really interested in, you know, how the heck do they make these giant blooms? So one hypothesis for how they make the blooms is that they are protected or armored by those coccolith plates that are around their body. So it's been sort of a long-held hypothesis that, you know, these organisms have evolved this protective mechanism. Grazers don't really want to eat them, and so they can just keep growing because they don't really have a loss process. And like a lot of things in science, sometimes things just get assumed but never tested. And so that seems to be the case here. And I became really interested in, well, like, we really need to test this. If we think that this is a protective mechanism, we really should test it. So that's what I set out to do. A couple years ago, I did an experiment where I fed a microzooplankton grazer different concentrations of either calcified or what I'm going to call naked E. hux. So that's just the body of E. hux without its coccoliths on it. And these are the results. So the way to interpret this graph is here on the x-axis is prey. So how much prey or how many E. hux I gave the predator in terms of carbon, and then the ingestion rate by the predator of those cells. And these numbers here aren't really important. These are strain numbers, different strains of E. hux. But what is important is the color. So dark or the black symbols are calcified strains, and the light symbols are my naked strains. And so hopefully what you can see is that there's not a super trend in between either the calcified or the naked strains, right? Here's a calcified strain that got ingested really, really well. Here's a calcified strain that didn't. Here's some naked strains, didn't get ingested really well. Here's one that did get ingested really quickly. So there's no clear delineation here between naked and calcified, which I thought was really interesting. So I looked a little closer, and I also tracked what happened to the grazers after they ate either the naked or the calcified cells. And here is the results of those experiments. So this here is prey abundance again, how much E. hux I gave them. And now we're looking at not the grazing rate, but the growth rate of the predator. So how well did it grow after it ate these different types of prey items? And so what you start to see here is an actual separation. So up here we see, especially at high concentrations of prey, when the grazers are fed the naked E. hux, they grow pretty well. But when they're fed the calcified E. hux, growth is really limited, almost to 0 or even below 0, which means you have mortality of the grazer population. And so this is really interesting. This sort of says that the grazers can't or don't discriminate between naked and calcified in terms of ingestion, but that there are ramifications for that ingestion after they do the ingestion, right? And so we're really interested in following this up. We have some experiments that are ongoing. We have tentatively dubbed this our Tums hypothesis because what we hypothesize is that as they're eating these really calcified cells, they sort of act like a Tums and neutralize a very acidic food vacuole where food and prey get digested. And if you're eating all of these calcified cells that have this calcium carbonate lifts on them, maybe you just stop digestion altogether and you can't grow. And so we're really interested in looking at the dissolution rates of calcified cells in the guts of these organisms to really sort of see what's happening. So if you take these grazing rates or ingestion rates and these growth rates of the predator, you can put them in a model. And this is a model. This is not something that I measured, but it's just a very basic model using the growth equation that I showed you a few slides back. And you can see what happens to an EHUX population over time. And so again, we have our naked strains and our calcified strains. And you can see based on the grazing rate of the ingestion rate of the predator and the growth rate of the predator, that naked strains are really readily consumed all the way down to 0 within 72 hours, so 3 days. And there's a lot of calcified cells that are still left. So even though there wasn't discrimination in terms of ingestion, there does seem to be some ramifications for eating calcified cells that may offer a level of protection to EHUX. Okay, so that's cool, that's in the lab, but what happens really in the field? Sometimes we do these lab experiments, they're not always translatable to the field, so we really became interested in what happens in the field as well. So what we did is we took a bunch of data from field experiments that myself and several other collaborators have done over the past 5 to 6 years. And we were able to separate out the phytoplankton groups in terms of size and also isolate EHUX. And using what's called the dilution experiment, I'm not going to get into that today, but it's basically a tried and true method that we have in biological oceanography to measure microzooplankton grazing. We could look at how grazers are consuming these sort of targeted groups. And we can compare, are they eating EHUX differently in the field than they're eating other phytoplankton that are of similar size, right? So we can't compare what happens to EHUX to what happens to maybe a giant diatom because they're too different in size to have sort of the same grazer try to attack them. So we need to really compare EHUX to something that's similar in size but not calcified. So here we have different types of phytoplankton. So here's EHUX here. This group is nanoeukaryotes. They're slightly bigger than EHUX, maybe between 10 to 20 microns in size. EHUX is around 4 to 5 micrometers in size. They're a little bit bigger, but somewhat close. We also have picoeukaryotes, which now are in the exact size range of EHUX. And down here we have Synechococcus, which is a cyanobacteria, which is about 2 microns, so a little bit smaller than EHUX. And this fancy U here, the mu, stands for the growth rate of these organisms, and the G stands for the grazing rate. And so what I hope you can see is that if you look across between what's happening on calcified EHUX cells and what's happening in a slightly larger group, an identical group, or a slightly smaller group, the only group that has significantly different growth and grazing rates are this smaller group here. That means that EHUX is growing at the same rate as these other very similar groups, but maybe more importantly, they're also being consumed at the same rate as these other groups, which indicates that there's generally no selectivity between these between EHUCs and these other non-calcified cells. You can also look at this in a different sort of way. This is called a Chesson and Manley index. It's a way in ecology that we look at feeding selectivity. An index of 0.25 indicates no selectivity. So that means that within the population of these different food items, predators aren't preferring one type over the other. And so what you can see is that between EHUCs, these nanoeukaryotes, and these picoeukaryotes, they really fall right almost on that 0.25 line, indicating no selectivity. And the only one that's different here is the Synechococcus. And so really we don't see any evidence of selectivity against E. huxleyi in natural populations, which indicates that grazers are really just able to chow down on E. hux, and that perhaps the coccoliths aren't as good a protector as we had originally thought. Now, we don't have the analogous measurements of the predator growth, and certainly that could be more impactful than what any differences in consumption would be. So we still need to explore that more, but in terms of just ingestion, grazers don't really seem to care about calcified cells. And so when we think about this in sort of more of a global context about what the implications are for not being protected against, using their coccolithophores as protection against grazing, we can think about how calcium carbonate is cycled through the ocean. So what generally happens is you have some interactions with CO2, you have calcification occurring. Usually these calcified cells are heavy, and so they also tend to stick together with other organic material in the ocean. They can form large aggregates, and because there are these sort of larger liths, larger in the terms of microbial world, they create larger sinking particles that conventionally have been thought to sink down to the deeper part of the ocean. They undergo a dissolution process, and upwelling cycles these nutrients and CO2 back into the euphotic zone. Sometimes calcium carbonate is deposited here on the ocean floor. Only about 9% that is exported from the top of the ocean to the bottom of the ocean gets preserved. And so this is how we sort of think that carbonate is cycled through the ocean. But if we now have grazers in here eating a bunch of calcified cells, it may be that dissolution is less likely to be happening here in the deep ocean, and a lot more dissolution is happening here in the surface ocean. And so this has the potential to change how much carbon now, how much calcium carbonate and carbon is exported into the deeper ocean. And we can get a better idea of how carbon is flowing through the ocean. And so that's sort of the importance of these investigations, to really look at these relationships on a really cool, intricate, zoomed in level, and then sort of take the results of those experiments and sort of zoom back out again and look and see what is happening over sort of large scale processes in the ocean. So the story for right now is that it looks like grazers don't really care about coccoliths and calcium carbonate, but only on the ingestion level. The digestion level is still an open story, and we're working on our Tums hypothesis. Okay, so I want to transition now to my second story. And this story is going to be using eDNA to really look at mortality and interactions between organisms. So this story starts in Georgia. This is my graduate student, Sean. He was a graduate student with me doing his PhD when I was working in Georgia. And his PhD consisted of doing grazing experiments once a week for a year off of our dock here in Georgia. And we also took samples to look at community composition using eDNA techniques. And I'm just going to briefly sort of walk through that process. We can talk more about that later if you want. But basically the process to look at eDNA starts with environmental sampling. So here Sean is taking a water sample. You bring your water sample back, you filter it, you extract the DNA from that sample. You use some specific primers and high throughput sequencing to sequence all the individuals in that sample. You do some bioinformatic processing, and then you get species identification. So you can see what are the individuals in that sample at that period of time. For our data set, we had 33 days over the course of a year, so about twice a month, we were able to have a sample. And we were looking specifically at the 18S rRNA gene. And so that will give us information about the eukaryotic cells there. We also have a story with the 16S rRNA gene that would give us the prokaryotes or the bacteria, but I don't have time to talk about that today. We're just going to focus on the eukaryotes. And so this is really a story of how you can use eDNA to not only tell you who's there, but to start to generate some hypotheses about what they're actually doing with each other while they're hanging out there. And hopefully what you can see is there's some familiar characters. So these are where the diatoms, this is the class of diatoms, are located in. And you know, these are pretty normal players for the coastal ocean. But what I do want to draw your attention to is this group down here, the Syndineales. And you can see relative to the other groups that we have, this group showed a really interesting seasonal pattern. About between June and October, they represented about 7 to 28% of relative abundance of the total population. And in the other months, they were either not there or they represented a relatively small fraction of the population. And we don't necessarily see that sort of seasonality with these other groups. And so we became really interested in sort of understanding what's happening with the Syndineales. And because this is a talk about mortality, you probably can assume or think about what Syndineales are. They're actually a group of parasites. So Syndineales are often the most abundant taxa in these sort of global 18S eDNA surveys. And this is the life cycle of Syndineales that most of them go through. So they can exist in this sort of dinospore stage where they can swim around just sort of like a small cell. They infect other dinoflagellates. They mature inside the dinoflagellate cell, and then they burst open, sadly killing clearly the dinoflagellate, and they sort of unfurl into this vermiform stage where these are essentially tiny dinospores all stuck together in this really creepy sort of worm-like structure, but also kind of cool. And then they eventually break apart and are dinospores and have to find a host to infect again. They have this cycle is pretty rapid, taking about 2 to 3 days. And each of these bursts can release, 100 or more dinospores per host. So they can be very prevalent in the ocean. And previous work has sort of shown that they can also be significant agents of mortality, specifically for dinoflagellates. So because we're super nerdy about mortality, we became really interested in this group. What are they doing? Who might they be interacting with? And so we asked a bunch of these different questions. How did this group change over time? What drives these changes? Who do they want to interact with? This is another image of an infected dinoflagellate. This is Akashua sanguinea. This is a bloom-forming dinoflagellate that commonly gets infected with the parasite. And this work has been published, so you can read more about our quest if you want to know more than what I'm going to talk about right now. So the first thing we wanted to know is, who are these organisms interacting with? And so we made a network analysis, and the way to sort of look at this network is to sort of use these branches and sort of let your eye follow them from one side to the other. So this large purple band here is all the Syndineales individuals that we found in our data set. And we wanted to see how they were connected to these other groups. And so blue connections mean they're positively correlated, meaning they occur together at the same time. And orange is that they're negatively correlated, meaning that they don't occur together at the same time. Now, I want to stress, just because they're, these parasites are connected with other things, either in a blue and orange way, it doesn't mean that that is a negative or positive interaction. It's just telling you when they either co-occur or when they don't co-occur. And you can use this data to start to generate hypotheses on potential interactions. So these don't describe or dictate interactions, but they give you some hypothesis fuel to then go and do further studies. So if you use that network then to calculate how things are connected to one another, what you can see are a few things. We see frequently observed infections or interactions with dinoflagellates. So they have a high, dinoflagellates belong in this group here. They have a high proportion of positive interactions, which means they co-occur together a lot at the same time. We also see sort of a higher proportion in ciliates. So these are microzooplankton that can get infected with parasites. And we don't really see a lot of interactions with abiotic factors. So also in this network, we tested different abiotic factors such as temperature or salinity to see if the occurrence of these parasites was more related to some kind of environmental factor. And we didn't necessarily see that. We saw more interactions with other types of organisms. And so what you can do is make a table that sort of looks like this. So here on the top, we have different individual hosts, and we have identified these as best as we could down to the species level. So we know what species of host this is. And then we have different species of parasites. These are different individual parasites that belong to different subgroups of parasites. And so what we wanted to do here is see how these groups interacted with each other, how these individuals now interacted with each other. So a couple things I want to point out. Take, for example, this Akashiwa sanguinea individual here. It was only related to one particular parasite, potentially meaning that this is a very close relationship between this particular parasite and this particular host. Whereas if you look across to other potential hosts, you can see that there's a lot of interaction or a lot of co-occurrence with a lot of different individual parasites, meaning this relationship may be more general. This host may be more of a generalist host than this host here, which has, maybe a tighter specificity for only a particular individual or type of parasite. And then similarly, you can see parasites that only infect a single host or only co-occur positively with a single host versus others that co-occur with many different hosts. So there's lots of interconnectivity here that's really interesting, and you can start to generate a lot of ecological questions about. Also really exciting to us, all of these organisms that have stars by them have all been confirmed to be infected with parasites in previously published work. So it's really encouraging to us that we see these potential hosts align very closely with these potential parasites. And so we can start to make some exciting hypotheses about what's going on here and how. And this is an example of that co-occurrence. So here again, we have our year data and our abundance here on the y-axis. And you can see the Akashiwa sanguinea, which is the host, and how it's related to and co-occurs pretty well with this particular individual group of, or individual parasite. And so we think that this is probably a pairing that is definitely co-occurring. We saw an Akashiwa sanguinea bloom during this sampling time period, and then it went away. It's known that this group is commonly terminated by parasites, and so this is something that we can dig into further. So future parasite directions, I am super excited about them, mostly because they occupy, when you do this eDNA work, time and time again, place and place again, they occupy quite a large proportion of the population. There's some technical caveats to why that might be, but there are also some really interesting ecological questions. And so those of you who have lived in this area for a long time know that historically the Gulf of Maine has been impacted by Alexandrium, a type of dinoflagellate that can cause paralytic shellfish poisoning. This is a map here of the 2019 cyst bed of Alexandrium. Alexandrium forms these resting cysts that can sit in the sediment, and every year some proportion of the population exist and can help facilitate bloom formation that way. This is the 2019 cyst count, so they use that to make a forecast about what 2020 will be. They forecasted a relatively low bloom year, and from what I know, that actually turned out to be true. But for the purposes of this conversation, I'm really interested in this parasite cycle that Alexandrium can have. So this is from a really nice paper that was done out on Cape Cod, where they looked at Alexandrium and in relationship to their parasites. And so there are a lot of outstanding questions just in terms of how these parasites interact with the dinoflagellates. We know that, you know, for terrestrial or other aquatic organisms, that temperature can be really impactful in mediating these relationships, and we're not as sure for phytoplankton, similarly with nutrients. And so there's a lot of, I think, really fascinating questions we can ask using this really important group from the Gulf of Maine. And so that's going to be some future work that we do in my lab. We're getting started on that this semester, now. And then also what I hope I can highlight for you is some ongoing and some interesting work with eDNA. So eDNA is sort of the up and coming, or probably already here, hot niche of oceanography. And it can be a very powerful tool. So here's another way of looking at our data set. And this time, these are different groups. And we looked at them over temperature, right? So we have a year-long data set from Georgia that spans a range of temperatures from about 6 or 7 degrees Celsius all the way up to about 31 or 32. And what we wanted to see is, how does the abundance of these organisms change over the changing temperature? So we can really track these groups that are really impacted or not by temperature. And you can see that depending on the group, there's sort of these different reaction patterns that arise. And so we're working on a network about this now and a paper about this now to sort of really understand how can we use eDNA to perhaps make sort of predictions about what will happen in a future ocean. So these are the eukaryotes. You can do the same thing with prokaryotes. I think this is really interesting here. This is SAR11, which is the most abundant bacteria on Earth. You can see that in this system here, it doesn't really seem, it seems to be super excited about warmer water. And so it's really interesting to think about, here's another SAR group that's sort of showing the same behavior. And so it's interesting to think about using this eDNA tool to answer a lot of different questions that we have locally about aquaculture, or patterns of phytoplankton, or patterns of larger organisms. We can start to use this sort of simple technology to really delve into how potentially climate or nutrient cycles or storms or all these other factors can impact the abundance and distribution of microbes in the ocean. Okay, so just to, in case you didn't get it until now, just to hammer the point home, the mechanisms of phytoplankton mortality is really matters in the ocean. And so really understanding how things die in the ocean is really important, right? We talked a lot about microzooplankton and that vectors carbon up the food chain. We talked about this sort of idea of the parasite loop. So that will infect phytoplankton cells, burst them open, allowing their nutrients to enter the microbial loop. Something we didn't talk about today, but is also very similar to parasites, is viruses. And I have a lot of ongoing work looking at when and how phytoplankton die from viruses versus microzooplankton and what impacts that has on carbon. And so I guess my take-home message would be that death matters and the type of death matters, and that to be able to really close the loop on how we understand sort of larger biogeochemical cycles, we really need to nail down these mortality processes. So with that, I want to thank you for your attention. This is not everyone in my lab, but some of the individuals in my lab. We were in Norway doing a really fun mesocosm project. I would like to thank my funders, and I will take questions. So thank you.
That was great. I love the Game of Thrones. As you were talking about that, I'm like, where's Parasite showed up in the Game of Thrones?
I know, not yet. Well, never, I guess it's over, but it would have been cool.
Cool. All right, excellent. We have some questions. And if I can, I just wanted to say one thing to Ingrid. I kind of answered your question, and I want to know if it changes your question that you want to ask Liz. Okay, so here's a good question from Denny. Do the grazers excrete the coccolith shells?
That's a really good question. For larger grazers, so you have to understand, like, everything in the microbial world is small, right? But for me, a larger grazer would be like a copepod, so a multicellular grazer. We know for copepods, they do excrete the coccoliths, but we don't know for microzooplankton. That's why we sort of are leaning to this Tums hypothesis that basically all of these liths just sort of gum up the works, essentially. The fecal pellets of microzooplankton are really small and are hard to distinguish from other detrital matter. So we don't quite know for microzooplankton.
I'm going to ask you a follow-up question to that, because I was thinking about the blooms. What causes the blooms, and do predators react to the blooms? And that's the place where maybe you could study that question.
Yeah, so we have done some experiments. This was actually this Norway adventure where we had these big, what are called mesocosms. Essentially, it's, you basically put a ginormous plastic bag in the ocean and cinch it closed, and you can do some manipulated experiments on them. And we stimulated a bloom, and we were actually looking at when viruses become important versus grazing. And grazing always seems to be important. Viruses really seem to be important in how the bloom demises. But how the bloom occurs in the first place, even with all this grazing, is probably a matter of being able to outcompete in the nutrients and light arena. And I don't think that's quite known 100% for EHUCs yet on how exactly they do it.
It's fascinating. It's amazing to think you can see all of that from space.
Yeah, for sure.
That's a lot of that organism. Okay, here's a question from Mike Ziegler. Could the prokaryotic temperature response for some taxa instead be due to seasonality? For example, more abundant in the summer, less abundant in the winter.
Yeah, for sure. It could for sure be because of some seasonality, that's intrinsic in there. I think really the power with the eDNA is to have your samples for, over multiple years and to really look at those trends over a multi-year scale. And so hopefully that's something, that we can get started and start doing to really sort of tease out those seasonal aspects versus what is really a temperature response. But it's interesting to think about and use that data to go back to the lab and do some interesting experiments or look more closely at this relationship in the field. So, but yeah, you're right.
What a great and sad experiment that you have in the Gulf of Maine that's warming every year.
I know, exactly, as we speak.
Wow. Well, and I also wanted to let you know that Shoals Marine Lab is going to start doing some eDNA sampling starting next summer.
It's very exciting.
So, maybe we can pick up some water for you.
Yes, I would love that.
All right. Well, that looks like that's it for the questions right now. Thank you for
Thanks, guys.
Those. And I just want to let everybody know that our next talk is next Tuesday, September 1st. Can't believe it's September. Same time, 7:30 with Daniel Pauly. He's actually probably one of the most famous fisheries biologists in the world. And he is going to be joining us. He, for those of you who don't know him, he established Fish Base, which is an encyclopedia of fish species, and EcoPath, which is a really well-known ecosystem modeling program that I have used. And he's the lead investigator of the Sea Around Us project, which looks at world fisheries and how to manage them. So he'll be with us next week, and you can find out all of this information and more about events we're doing and talks that are coming up. We're going to be doing these rock talks until the end of December. So please stick with us. There's so many great talks coming up. So thanks for joining us. Everybody take care. Thank you, Liz, so much. That was great.
And have a good night.
Take care, everyone. Good night.